Hello, I'm Nick Burton. I'm a research fellow here at Pembroke in genetics. I predominantly work on how a mother's environment affects offspring. And the reason I'm particularly interested in that is that there are, in fact, a number of rather amazing examples in invertebrates where a mother's environment can have profound and dramatic effects on offspring development and physiology. For example, one of my favorite examples is in pea aphids, where a mother's exposure to a variety of different environmental stresses can be sensed, and she can program her offspring to develop wings when they otherwise wouldn't. And this is a sort of example of what I call maternal bet hedging, where the mother senses the environment and sort of programs her offspring to have the best possible chance at survival and reproduction in whatever environment they're about to face. But the reason it's referred to as bet hedging is that it often comes at the expense of the offspring's ability to respond to mismatched environmental conditions. So these types of effects have been observed obviously in invertebrates and in mammals now, and what my research proposes is that they are in fact more common than previously anticipated, and this is just a sort of universal strategy to adapt to changing environmental conditions. In addition, I sort of propose that this type of mechanism or related mechanisms either underlie or contribute to a variety of human pathologies that are linked to a mother's environment. So one of the better examples of that is that low birth weight is strongly correlated with development of type 2 diabetes or cardiovascular disease in later life. And low birth weight can also be caused by maternal exposure to environmental stress. And that could just be deleterious consequences of environmental stress affecting the offspring in negative ways that results in the development of these diseases in later life. Alternatively, based on what we know about invertebrates, it's possible that the low birth weight is an adaptive response to environmental stress in utero. That comes at the expense of long-term health. So it ensures proper development in conditions where resources are limited, but it comes with these long-term consequences as an example of maternal bet hedging. And that sort of remains unknown, but is a topic that I'm very interested in. And so to do this research, I work on a roundworm called Caenorhabditis elegans. It was sort of made famous here at Cambridge as a genetic model organism, and many significant molecular pathways were discovered in these roundworms that are conserved all the way through humans and regulate similar physiology. For example, the insulin pathway is one that most people are familiar with, and a lot of really seminal work on insulin signaling was done in C. elegans. And what I work on is how C. elegans and that roundworm adapts to osmotic stress, which is just salt stress. These roundworms have trouble dealing with seawater, so roughly conditions of seawater levels of salt stress. And if these embryos are exposed to this salt stress, they will arrest their development and enter the state of what resembles suspended animation to cope with this environmental stress. However, if you expose their mothers to a mild version of salt stress, this can protect her offspring. And that protection results in offspring that bypass this arrest or suspended animation and can develop even at sort of sea salt levels of osmotic stress. And I work on how does that mechanistically happen. And what I found is that it's in fact regulated by insulin-like signaling to a variety of tissues, most significantly insulin signaling to the mother's germ cells or oocytes, which programs the offspring to produce more or less glycerol, which is something that can protect them. It's just a molecule that can protect them from the effects of osmotic stress. And so what I now work on is how does insulin signaling result in a change of information that affects the offspring metabolism. And what I hope is that this will uncover some mechanisms that are important also to humans, where we see maternal stress affecting offspring metabolism and physiology. We know insulin signaling is well conserved throughout animals, and so I propose that insulin signaling to mammalian germ cells, so a mother's oocytes, is also programming offspring physiology in mammals, and that's something I hope to test here. I hope to expand into mammalian work and generally work on how do environmental stresses communicate across a generation, if that's at all possible, and how does mismatches in environment affect growth and development in physiology.